Hello, today we're going to be talking about metabolism part 2, and specifically cellular respiration and fermentation. So, chemical energy in food. Food provides living things with chemical building blocks to grow and reproduce, and a source of energy. How much energy is actually present in food? Well, in 1 gram of sugar or glucose, which is C6H12O6, it releases 3,811 calories of heat energy. Now, a calorie is the amount of energy needed to raise the temperature of 1 gram of water 1 degree. It's just a unit of measure. And the calorie is going to be a capital C, and it's used in food labels as kilocalories, or 1,000 calories. Now, we're going to be talking about cellular respiration, so let's take an overview. Glycolysis is followed by the Krebs cycle, and then the electron transport chain in the presence of oxygen. Cellular respiration is the process of releasing energy by breaking down glucose and other food molecules in the presence of oxygen. Again, oxygen is a key piece here. It's going to be the exact opposite of photosynthesis, except we don't need light, and we have ATP added here at the end. So it's going to be 6 oxygen plus glucose yields 6 carbon dioxide, 6 water, and energy, such as ATP. Yay! Okay. 3 main stages. We've got glycolysis, the Krebs cycle, and then the electron transport chain. They take place in the cytoplasm and in the mitochondria. So, stages, again, first stage is going to be glycolysis, and it's anaerobic. It does not care if it has oxygen or not, so it can be done in both aerobic or anaerobic, because no oxygen is actually required. Now, the last 2 stages, the Krebs cycle and the electron transport chain, are aerobic, and they require oxygen to be completed. So looking at this graphic representation, cellular respiration is going to be on this side. Now we can do fermentation, and we'll get to that later. But we have glycolysis occurring, and then we have our Krebs cycle, and then we have our electron transport chain. Again, cellular respiration occurs in the presence of oxygen. Glycolysis. Glyco meaning sugar. Lysis meaning to cut. So glycolysis, we are cutting sugar. It occurs in the cytoplasm, the process of breaking down 1 molecule of glucose into 2 pyruvic acids. So, in this whole thing, it produces 2 net ATP, 2 net energy. Yay! 2 pyruvic acids that are going to be used in the Krebs cycle, or for fermentation, and then 2 NADHs that are going to be used later in the electron transport chain. Krebs cycle comes after glycolysis. We're going to take those 2 pyruvic acids and break them down into energy extracting reactions, Oxygen is required. The pyruvic acid is transformed into acetyl-CoA and breaks down to form ATP and CO2, or carbon dioxide. Now, the Krebs cycle goes around twice. So for every 2 turns, we produce 2 ATP. Yay! Then 6 NADH, 2 FADHs, which are molecules that are going to be used as electron donors in our electron transport chain. Now, comes next to the electron transport chain. It takes place in the inner membrane of the mitochondria. So we've moved from the cytoplasm into the mitochondria. The electrons from breaking of the 6 NADH and the 2 FADHs and the other high-energy molecules from glycolysis are passed through the electron transport chain along the electron carriers, as we saw in photosynthesis. Every time it jumps, it releases a little bit of its energy, and it pumps a proton, or an H+, into the internal membrane of the mitochondria. This forms the electrical or chemical gradient, also known as the proton motive force, and the proton gradient, as it becomes more positive inside of the mitochondria and more negative outside of the mitochondria. Now, as 3 H's then go through the ATP synthase protein, it provides enough energy for 1 ATP to be created. ATP. Yay! Okay, again, this is ATP synthase. This molecule actually turns as a proton, or an H+, goes through it. So it's sort of like a rubber band. So it just sits here, and as 1 H plus goes through, it winds it around, and then another one goes through and winds it around again, and a third one goes through and winds it around. Then it has enough energy to create 1 ATP. That energy is needed to combine ADP and PI. So the electron transport, the final electron acceptor is oxygen. If it's not oxygen, it does not work. And this forms 2 molecules of water. So total in the electron transport chain, we have 36 ATP, and we have 2 H2Os, or 2 waters. So, overall, glycolysis, we get 2 ATP. Die! Krebs cycle, we get 2 ATP. Die! And then we get 34 ATP with the electron transport chain. That gets a double. Die! Die! Break down again. Total of 38 ATP when oxygen is present. That is awesome. Now, what happens when oxygen is not present? During fermentation is when oxygen is not present. You go through glycolysis to create the 2 ATP, and then you have the pyruvic acid. Now, the pyruvic acid is then broken down into different forms, and let's take a look at those different forms. It occurs when no oxygen is available. It's an anaerobic and follows glycolysis, releases energy from food molecules to produce ATP in the absence of oxygen. 2 types. Alcohol fermentation, which means the pyruvic acid is turned into alcohol, and then lactic acid fermentation, which means the pyruvic acid is turned into lactic acid. Alcohol fermentation, mostly used by yeast, produces CO2, ethyl alcohol. It combines the gas in the air pockets of bread, and so that is the smell that you smell when you are cooking bread. It's most of the time the alcohol and the CO2. And we take the pyruvic acid, and we come out with alcohol and CO2. So that's going to be alcohol fermentation. Lactic acid fermentation occurs in us, and it's the process of converting the pyruvic acid into lactic acid. Pyruvic acid to lactic acid. Now, all my athletes out there, you start running, you're doing good, and then all of a sudden you start feeling this burning in your muscles. Well, when you're feeling good, that beginning portion, you have a lot of oxygen built up in your muscle cells, and your cells go through cellular respiration. But when that oxygen runs out, you start performing lactic acid fermentation to still create that ATP needed. Now, the lactic acid builds up in the muscles, which actually causes that burning feeling and that fatigued feeling and the sore muscles. So, the longer that you can go without actually starting lactic acid fermentation without that burn, the better shape you are in. So again, fermentation energy total, glycolysis, 2 ATP. Die! Fermentation, none. So, this type of energy creation is not very efficient. We would rather do cellular respiration and get 38 ATP. But sometimes we don't have enough oxygen, so we have to go this route that produces 2 ATP. Okay, cellular respiration breakdown uses glucose, or glycolysis uses glucose, produces pyruvic acid and 2 ATP. The Krebs cycle and electron transport chain, when oxygen is available, creates high energy molecules and produces 36 ATP and oxygen. Fermentation, when no oxygen is available, creates 2 ATP, pyruvic acid, and then goes through either alcohol or lactic acid fermentation to produce alcohol and lactic acid. Now comparing the two, photosynthesis versus cellular respiration. Big thing, photosynthesis creates the food. The energy is stored in glucose. Creates the food stored in glucose. Cellular respiration, it is now breaking down that food, breaking down that glucose to create ATP. Quick energy. Photosynthesis takes in carbon dioxide and gives off oxygen. Cellular respiration gives off CO2 and takes in oxygen, so it's reversed. Photosynthesis only goes in the light because remember our photosystems must have light, and chlorophyll must be present. For cellular respiration, it can occur in both light and dark, and all living things, including plants, do cellular respiration. So keep smiling, eating, and breathing, because you need all of those for happy, optimal energy production.